Diagnosis of bovine theileriosis. Bovine theileriosis is a disease caused by a blood parasite which called Theileria species. There are two common types of Theileria species parasites, including Theileria parva and Theileria annulata. This blood parasite is transmitted to cattle through biting of infected ticks. The most important clinical signs of the disease including lacrimation, nasal discharge, high fever which may reach 41 to 42 degrees centigrade. One of the most important characteristic clinical signs is the enlargement of the superficial lymph node, especially prescapular and prefemoral lymph node. The second important clinical sign is the presence of ticks all over the animal body, also in the perineal region. These two areas, the area under the tail and the perineal region, is considered the prediction seat for the ticks, as this area the skin is thin and containing less hair, which make it easier for the ticks to suck blood from the animal. Here in this case, there is a bilateral enlargement of the prescapular lymph node and also enlargement in the prefemoral lymph node. The sporozoid is transmitted from the infected ticks to the animal, reach to the lymph node, and then multiply in the lymph node and result in the enlargement of the lymph node. And form the schizont. After that, the sporozoid invade the red blood cells. So the presence of these clinical signs of the animal suggests the infection of the animal with the Theileria species parasite. To confirm the presence of the Theileria species parasite, the second step of the diagnosis must be made, which is the laboratory diagnosis. Laboratory diagnosis of bovine theileriosis is based on a simple method, which is examination of a stained blood film. The blood sample can be collected on a EDTA tube containing EDTA as anticoagulant, and then a blood film is prepared directly after collection. Or the blood film can be prepared directly by a drop of blood obtained directly from the ear vein of the animal. To prepare the blood film, first mix the blood sample gently. After that, spread the blood film by using the two slides method. A drop of blood is placed in one end of a glass slide, and then the blood film is spread by using another glass slide. After spreading the blood film, allow the blood film to dry, and then identify the blood film by labeling the thick end of the film. It is better to make more than one blood film to permit thorough examination. After dryness of the blood film is fixed in absolute methanol or absolute ethanol and allowed to stand for 3 to 5 minutes. After fixation of the blood film is removed from the alcohol and then allowed to dry. After dryness of the blood film is stained in Giemsa stain. Giemsa stain used here is a diluted Giemsa stain, 10% Giemsa stain solution, and the blood film is allowed to stand in the stain for about 40 minutes. After that, the blood film is removed from the stain and then washed with using buffer or distilled water, and then one drop of oil is placed to one end, to the thin end of the film, and then examined under the oil immersion lens. The Theileria parasite is a intracellular blood parasite present inside the red blood cells and takes the form of signet ring. So the presence of signet ring blood parasite inside the red blood cells is characteristic for the Theileria annulata infection. Here in this blood film, here we found that more than one red blood cells infected. Here red blood cells infected, red blood cell infected with CD parasite at the same time. Here another blood parasite Theileria present inside the red blood cells, which takes the form of signet ring blood parasite, which means that the animal is positive for Theileria species infection. Also, during examination, notice the red blood cells. The red blood cells is larger than normal, which may indicate the presence of reticulocytosis and indicate the presence of anemia. This is another blood film. Here there is two red blood cells infected with Theileria annulata parasite, which is signet ring blood parasite. Also here, signet ring blood parasite present inside the red blood cell.